Thank you very much for the invitation. I was told to be fast, to hurry up. I don't know the right person to ask for that, but I will do my best. So I will talk about structural biology of neurodegenerative diseases, and mainly focus on Parkinson's disease. As you can see here in this slide, there are several evidences that link the etiology of Parkinson's disease and the development of Parkinson's disease with the amyloid aggregation of the protein alpha-synuclein. And also you can see there that these evidences have been reported very recently, so we can conclude altogether that this, by sure, is an active area of research. The protein cell, as Christian and told already told, is a small protein, 140 amino acids, distributed in 3 well-defined regions. The N-terminal region that is amphipathic, the N region that is hydrophobic and is related to the amyloidogenic capacity of the protein, and the C-terminal region that is very rich in negatively charged amino acids and acts blocking the aggregation or the amyloidogenicity of the protein. The physiological role of the protein is not well understood yet, and the protein belongs to the family of intrinsically disordered protein. It means that the protein lacks rigid elements of secondary structure, and the protein lacks well-defined tertiary long-range contacts. And indeed, the protein cannot be crystallized. So the only source for study the structural and dynamic property of the protein is by nuclear magnetic resonance. In this kind of protein, we cannot talk about structure, but we talk about the propensity to adopt some kind of a structure or a structural propensity. The aggregation process of the protein is nucleation dependent. You can see here there are several factors and effectors that influence the aggregation of the protein, and in our lab we mainly work in the transition that takes place from the monomer state of the protein towards the aggregation prone state. Once this structural transition takes place, the aggregation process is triggered and you cannot stop. So we try to understand which are the structural factors involved in this transition that takes place at the very early stage of the amyloid aggregation process of the protein. As you can see in this slide, if you compare the target protein involved in several or in different neurodegeneration processes, as I mentioned, the only protein that is not that lack well-defined structural element is the protein involved in Parkinson's disease and is alpha-synuclein. That's why the protein cannot be crystallized, cannot be studied by X-ray crystallography. So we designed a strategy together during my stay in the laboratory of Tom Chauvin with the NMR basic department of Christian Giesinger, and a strategy that involves to know or to elucidate the structural and dynamic features of alpha-synuclein by using nuclear magnetic resonance. I will talk today about a research line in my group that work trying to understand the protein-ligand interactions. As you can see, there is key aspect of these diseases that are not well understood. So, but there is a clear relationship between the amyloid aggregation of the protein involved in neurodegeneration and the toxic effect on neurons. So there is a strategy that involves the discovery and design of small compounds that tries to interfere with this process here in the aggregation pathway of the protein. However, even if there is a lot on several compounds that has been assayed, and even several of them are being used in clinical trials, there is not knowledge at all, or it is well not understood, I decide to be more flexible, about the structural basis and molecular mechanism behind the inhibitory effect of these compounds on the amyloid aggregation of the protein. And if you want really to go and advance in the design of new compounds, I mean more effective compounds, you need to count with this information. You have to know this information, which are the target site in the proteins, which are the structure of the complexes that are formed, and which is the molecular mechanism by which these compounds inhibits the amyloid aggregation of the protein. So we started with just to work with one of these compounds in the laboratory that is a tetrapyrrolic compound. The compound is phthalocyanine tetrasulfonate. That is a It's a polyaromatic compound that has negatively charged sulfonate groups in the periphery, and this compound has been shown to be neuroprotective against Alzheimer, Parkinson, and prion disease. And as you can see here in this electronic micrography, the amyloid fiber formation is strongly inhibited in samples of the protein that are incubated in the presence of these compounds. And it was proposed and even published that the inhibitory effect of this compound was involved the interaction with the C-terminal region in the protein. However, in some of the previous slides, I show you that the C-terminal region of the protein is very rich in negatively charged residues. So if you just take a look at the periphery of this compound that is also has attached negative sulfonates, you can understand that instead of a binding, you expect to see a repulsion. So we started to study the interaction and to try to identify the ligand binding interface for these compounds in the protein. And this is intensity profiles or interaction profiles that in which you demonstrate how the intensity of the signal change in the NMR spectra upon the presence of increasing concentration of the ligand. In that way, if you make an interpretation of these effects in function of the primary sequence of the protein, then you can easily identify the binding interface of this compound in the protein. And you can see here there is two main sites of interaction with this compound, and that these sites are in the N-terminal region, which is absolutely opposite or in contrast what was published before, that it was supposed to be here, the interaction to take in the C-terminal region. So this is very important to know this, because this is very important structural information that has to be or can be used to design more efficient amyloid inhibitors. In particular, we noticed that all these affected regions, even also there is a weak affected region here, was centered on amino acid with aromatic side chains. Then we started to prepare mutation. We use this information to make a rational design of different variants of the proteins, in which we made a point mutation of this aromatic region and to see what happened. And you can see here in this mutant, when we mutated the tyrosine at position 39, there is this binding component is loose, okay? And when we mutated phenylalanine at position 4, there is just a residual component of the binding. So we can also build a ranking for the affected region by this compound, and from this ranking that is built according to the degree of broadening induced by the compound or the resonances of these residues, you can see that the residue located at position 39 resulted to be the more affected. This region containing this residue is being the more affected by the presence of the compound. So we wonder, is this the target site through which this molecule inhibit the amyloid aggregation of the protein? Then we decided to, the next step, to evaluate the amyloidogenic potential of these variants that we have characterized structurally. And you can see here that in the case of the variants, these variants in which the phenylalanine was mutated at position 4, these mutants were able to form amyloid fibers with the same morphology that the wild-type protein. But in those cases where the mutation was around involving the tyrosine at position 39, there was just oligomers were formed, and there was no formation of amyloid fibers anymore. So it means we can conclude from here that the tyrosine 39 seems to play a critical role in the fibrillation pathway of the protein, and that by sure this is the residue that is being targeted by the PCTS compound to exert its inhibitory effect. I mean, there must be something else stabilizing the complex or the interaction between the aromatic side chain as these polyaromatic compounds, because otherwise, according to this effect, we expect that the dissociation constant is around in the low micromolar range. So probably there is a stabilization. This complex is stabilized by the electrostatic contribution coming from lysines located around the tyrosine 39, or maybe some residual structure that could be around this residue. So, which is now the molecular mechanism by which this compound inhibit? We know which are the molecular, the structural bases that involve the interaction, probably p-stacking interaction with aromatic compounds, and the mechanism may involve two processes. Now is the interaction or influencing the on-pathway oligomers, or redirecting, making a redirection of the pathway towards an off-pathway mechanism that give rise to oligomers that are completely different in terms of the structure morphology to that involved in the on-pathway mechanism. And you can see here, we combine this with biochemical studies that involve the use of the soft detergent sarcosine that dissolve intermediates but not fibrils. Then you can see that the very early stage of the aggregation of the aggregation process, there is an accumulation of prefibrillar species of the protein. So it means prefibrillar accumulation of, it means that the off pathway is not operative, so the interaction may involve the trapping of prefibrillar alpha-synuclein species, influencing or retarding their evolution to the formation of amyloid-like fibrils of alpha-synuclein. So schematically, okay, so this is the mechanism. As you can see, there is interaction of small molecules with the protein. Then when the proteins start to form the first nuclei, probably the large oligomeric forms started to be formed, but they cannot be promoted or to evolve towards the formation of amyloid fibrils. So this is the intermediates that are being accumulated and that we are detecting. So this is the mechanism involved in the interaction, in the inhibitory effect displayed by this molecule. So we have described the structural basis behind this mechanism and the molecular basis behind this mechanism. Now we have very well detailed information to go ahead with the design of more efficient amyloid inhibitor, but we really want to go to understand even better this process. So one of my students in my lab prepared different mutants of alpha-synuclein to test if the position, the hydrophobicity of the aromatic character of the residue at position 39 was the main driving force, was the key factor directing the aggregation pathway of the nuclein. And you can see when that in this slide that for the free proteins that these mutants in which the hydrophobicity is almost conserved because we have a mutation from tyrosine by leucine or isoleucine, then you see that the proteins in the free state are able to form amyloid-like fibrils. So it seems to be that the hydrophobic character of the amino acid at this position is key for the fibrillation of the protein. But contrast to that, the interaction profile when you use these compounds, PCT, is completely different. And in this case, this compound is not able to inhibit the amyloid fibril formation of the protein. So the aromatic character of the tyrosine at this position, the residue at this position, is key for the anti-amyloid effect of this compound on alpha-synuclein. If this aromatic side chain is not there, so this compound is not functional. I mean, it's not active as an inhibitor. So we studied the interaction of these compounds with the A-beta peptide and to see if this compound is able to inhibit also the amyloid fibril formation of A-beta peptide. A-beta peptide is characterized, in this case, we studied the 1 to 40 amino acids. The first 15 amino acids are very flexible, very disordered, and they have a tyrosine there and 3 histidines there. And you can see there is this flexibility allows the contact between these two regions, between 17 and 20 and 31 to 36, that are important for the amyloidogenicity of this molecule. Then when we studied the interaction of this compound with alpha-synuclein, we detected a main interaction site around tyrosine 30 and also some weaker interaction that was driven by hydrophobic interactions. In that case, when we studied the amyloidogenic potential of the A-beta, it was not the amyloidogenicity was not inhibited by the interaction of the compounds. And indeed, the interaction of these compounds was very weak compared to alpha-synuclein. You see So, okay, since there's no inhibitory effect displayed by this small molecule on the amyloidogenicity of the interaction profile suggested us that this may be due to the weaker interaction between these compounds and the beta molecule. And then if you compare the interaction profiles, how the interaction profile of this complex change upon the presence of unlabeled alpha-synuclein, you see that when you add increasing concentration of alpha-synuclein, you recover the NMR profile of the free A-beta molecule. It means that there is a complete release of the compounds from the complex formed with A-beta. It means that the interaction is much weaker between the interaction between PCTS and A-beta amyloid is much weaker than those involved in alpha-synuclein. But then, since the flexible region of the first flexible region of A-beta peptide is very important to modulating the amyloidogenic capacity of this molecule, we designed a new compound in which simply we coordinate a zinc ion to the central cavity, and zinc has high affinity to coordinate for histidine residues. And you can see here the interaction profile changed substantially in the presence of when zinc is coordinated to this inhibitor, and when we analyze the amyloid fibril formation of this compound, of this molecule in the presence of this compound, there is a total inhibition of the amyloid fibril formation by the interaction of this compound to a beta molecule that now is driven mainly by the interaction or the coordination of zinc to the 3 histidines there. And in that case, the coordination of this molecule through the zinc ion to these regions make that this molecule gains rigidity, is more compact in the region. So these contacts between these two regions are not so strong, so the amyloidogenic potential of this compound, of this A-beta molecule is strongly decreased. Finally, I just want to say a few words, because I have shown you a lot of results, and results that were developed by my group here in Rosario. I just came back in 2006 because there was an investment in science and technology field. There was the Agency of Promotion and Technology at this period made an investment, and they bought the first high-resolution NMR instrument in the country. And you can see how this is reported in a journal that a unique laboratory in the country was created in 2006. The problem there was only a unique investigator, I mean, only me and the magnet. So we need to perform a strategy to, try to imitate what happened in the NMR-based structure department headed by Christian Griesing, and I have the luck to enjoy during my stay in Germany. So we started to design a strategy to create a school of NMR structural biology in Argentina. And one of the points of this strategy was the creation of a partner group that to develop joint research projects between my group in Rosario and the group headed by Christian Griesing in Göttingen, and also to create education opportunities that link the University of Göttingen with the University of Rosario. And we are working that in creating a joint PhD program in which students from both countries may be involved, and they could get a double diploma. In that was also very important the University of Rosario, the University of Göttingen, the Max Planck Society, and in particular the Max Planck Institute for Biophysical Chemistry, and the Alexander von Humboldt Foundation. So with this funding and with this support, we were able to perform in Argentina a system that allows us to change students between labs and to organize a series of workshops and courses to train the students, because you can imagine the cost of the instrument was $1.2 million. So the students at my lab were very scared just to press, okay, to perform experiments. So we organize a course per year in the period 06-09. We also organized 2 bilateral workshops in 010 and 011. I mean, several students all over the world came to Rosario. Rosario began to be, became a center of reference in NMR-based structural biology in Latin America. And by supporting of all these tools, we went from this original situation to this original situation that is right now. So it is important to show you that when the interaction, there is interaction, and the interaction is serious and well designed, though it's possible to show and to make deep changes even in short times. This interaction just is running in the 6th year. And I have to tell you that Professor Christian Griesinger, that just gave a talk here, have supported a lot to us, and he's the main contributor, I can say, for the development of nuclear magnetic resonance, not just in Argentina, but also in Latin America. He's doing also effort in Brazil and Colombia. Okay, I can tell you that Christian has written several chapters in the story of the NMR development, I mean, NMR of biomolecules, in the development of NMR biomolecules in our country. As can you see here, this is, we have published, I almost finished. We have published 21 publications in peer review and prestigious international journals. And from that, you can see that 50% of the work that were published were performed entirely in Argentina and were signed as correspondent also by me. It means that this is real interaction.